And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Awesome. Okay. Yeah, looks like we're live. Guys, it's Standing for Truth. We've got a good discussion tonight. We've got David here. His YouTube channel is Kakarot. Those Dragon Ball Z fans will understand that one, that's for sure. This isn't David's first rodeo. He's recently had a debate with Kent Hovind. It was more of a formal debate on modern day debates. Really respectful dialogue, so that's why I invited David here today. So it's going to be more laid back, more discussion based, but we're going to give David here plenty of time to go over his position, make his points. Before we even get into the discussion, David, I'm going to throw it over to you if you want to just give a brief introduction, if you want to plug your channel, let everybody know what you're doing at your channel, if there's anything you wanted to say, and then we'll kind of get more into it. So yeah, welcome, David. Thanks for joining.
Yeah, thanks so much for having me. Yeah, I'm so happy that you invited me. It's awesome. Yeah, I've been wanting to talk with you for a while now. So thanks so much for inviting me.
Awesome. Yeah, no, I'm excited too. We love to get people of opposing views that are interested in respectful and profitable dialogue. So if you're looking for more people to check out your channel and the videos you're putting out and stuff as well, I'll put your channel in the description box.
Thanks.
And for anybody who's listening, if they like what they're hearing regarding David here, go click like and subscribe. So why don't we get right into it? The discussion, the topic's going to be more so, we'll try and keep it on topic as much as possible, but I mean, sometimes it's inevitable to drift off a bit, but universal ancestry or separate ancestry. Everybody knows our positions. Obviously, I'm going to be defending independent ancestry according to the biblical base model, and David's going to be defending universal common ancestry. So why don't we get a little more details on your position, David? I'm going to hand it over to you. Take as much uninterrupted time as you need. If you need a few minutes, that's fine as well. If you ever need to share a screen or anything as well during the broadcast, let us know, and Praise will be more than happy to work that out. So I'll hand it over to you. Take a couple minutes, whatever you need, and just kind of go over your position, David.
All right, thanks. All right, so for starters, I think what mostly convinces me of universal common ancestry is that pretty much all life is based on DNA. And DNA is, used to trace, our hereditary relationships with other people. They use paternity tests. They use DNA to, determine, if I'm the father of this child or whatever. Yeah, my dog's, barking.
That's okay.
They use the exact same type of tests to determine our relationships with other organisms, like, for example, the great apes, as they do to determine our relationship with another person. So that's pretty overwhelming. Also to top that off, there are also some transitional fossils that we found that are kind of in between what most creationists that I know would consider to be different kinds. So like dinosaurs and birds. So like, for example, Microraptor and Archaeopteryx. Those are a mosaic of, different kinds. And also Homo erectus. That's another good example. Especially depending on the specimen. A lot of creationists, when looking at, I believe, Java Man, some will say that it's just an ape, and others will say it's just a human. So that's a pretty good indicator that it's in transition between what we think of as apes and modern humans. And also embryology. When you look at embryos of sorry about that. When you look at embryos in different stages. The earlier they are, the more similar they look to each other. So I think that those are some of the most compelling. But also another thing is the geographical distribution of organisms. So, like, for example, Australia is a safe haven for marsupials. They're all very closely related there to each other. They're all pretty much all the mammals there have pouches. And that makes sense given that it's been isolated from the rest of the world. So those are just a few examples of why I'm convinced. So, yeah.
Awesome. Yeah, no, I think I appreciate that. So I guess I'll briefly go over my position, and then we can kind of touch on as many of those things as possible. So from what I can remember, you said transitional form, specifically your theropod dinosaur to bird transition, genetics, paternity tests, for example. So I guess just a number of genetic related lines of evidence. Embryology, and what was the and then the geographical distribution of organisms, marsupial, for example, in Australia. And you mentioned some of the so-called pre-human fossils, like Homo erectus, for example.
Right.
Are there any other ones you would you say erectus is one of the more stronger hominids that you'd look at, or is there any other ones you want to put in there?
Absolutely. Yeah. Homo habilis, Homo erectus, Homo heidelbergensis, also Australopithecus afarensis. Those are some of the primary ones. So,
right.
Yeah.
Okay. Yeah, no, I appreciate those lines of evidence, because to be honest with you, I think that will help us have a good back and forth discussion. We got lots to talk about. My position briefly, which would be a limited ancestry. I think, and I agree with you on what you said at the beginning, that it's genetics, it's genes, it's traits that are inherited sperm and egg, right? A rock, for example, is not inherited sperm and egg. Geography is not inherited sperm and egg. And these are all good things to discuss and to discuss in relation to our models. But the only direct way of determining ancestry would be in genetics. So I do agree with that. So I'm looking forward to discussing those, everything you said, but especially focusing on genetics, because I think according to our model, we can look at, for example, just the genetic structure in humans overall. I think that speaks to us of a literal Adam and Eve. So oftentimes evolutionists, and even with R and Ron, his phylogeny challenge, they'll say, give us a specific kind. And I think based on molecular clocks and based on DNA function, for example, which we can get into, has determined that humans are their own separate kind, humankind, for example. So we can go over those lines of evidence as well. We would look to just the empirical method, pedigree-based mutation rate studies, for example, in the Y chromosome and the mitochondrial DNA, that takes us back just to 6,000 years for mitochondrial Eve. And for Y chromosome Adam, it looks like the Y chromosome actually mutates faster than we thought. So it actually takes us back about 4,500 years to Noah at the flood, which is fascinating. And I know the evolutionists, they'll look to phylogeny-based assumptions, the fossil record, to calibrate those dates with their assumed dates of deep time evolution. So we can discuss that as well. I understand that they have their own, I guess, interpretation of that. When it comes to the dino to bird evolution, and we can discuss this too. I'm not sure if you're aware of the modern bird tracks, although I believe the secular evolutionist side will say that these are more so proto-bird tracks. But these tracks are found going back 200 million years ago. I believe these fossilized bird tracks are found in the late Triassic sediments, when evolutionists believe the very earliest dinosaurs were supposedly extinct, supposed 55 million years before Archaeopteryx came along, for example. But with Archaeopteryx, we can look at Archaeopteryx Tiktaalik. We can look at the reptile mammal transitions, for example, those mammal-like reptiles that seem to be highly varied, and actually they were a widely distributed group of reptiles that R.J. Downard has written a book on this, but they had what seems to be a number of characteristics that are found in mammals. But when it comes to like mosaics, for example, or that which is transitional, our model is based on design, for example. So the Bible says, and I'll just go over this briefly, the Bible says that we're made in the image of God. Therefore, we should be able to get a sense for how God may have designed life, created life, by the way we design things, for example. So, I mean, we could look at many things, but if we just look at design modes of transportation, they just so happen to fall within these groups within groups patterns, nested hierarchical patterns, but also regarding the transitional forms, it just so happens that we design things that seem to blend the features of two different types of vehicles. For example, in the military, you've got military amphibious assault vehicles, which are built for the transition between land and between the ocean, right? So these amphibious assault vehicles, they exist, and they're blending the features of, like I said, land vehicles and water vehicles. So we can also look at crossover SUVs. So according to our model, we would expect some type of mosaics. It seems like the majority does prove the rule, though. I mean, there are a select few that we can talk about. And there are mosaics today, too. I mean, you can look at the living duck-billed platypus. That creature is a mammal, and yet it has a duck bill, webbed feet, lays eggs, and in addition to possessing other characteristics that one could even call, let's say, reptilian, it actually has characteristics of mammals, reptiles, and birds. But the point is, yeah, we see mosaics. We see mosaics in the design world as well. Therefore, it's not a surprise for us to find those types of creatures. But a lot of those, especially in the fossil record, unfortunately, there's no DNA to go with them. That's why, like I said, it's DNA that's inherited, sperm and egg, not a rock, not geography. You mentioned a couple other things, but I don't want to go on for too long. But I'll throw it over to you if you want to give a few thoughts on what I said, and I'll let you take the conversation in the direction that you so think is best. Go ahead, David.
All right, thanks. So you mentioned about bird tracks in the late Triassic period?
The late, so there's a paper, actually, I think it's actually been retracted, as in so the late Triassic period, yeah, that's initially what they said, which is a problem for dino to bird evolution. Now I believe they're trying to say that it's the late Eocene. I think they've recalibrated the dates, but yeah, I've got a paper that suggests that. Have you heard of that one, David?
I don't know. I can't say that I've heard of birds appearing in the Triassic. That's certainly a possibility. I've just never heard of it before. So,
yeah, it looks like the tracks are so clearly bird-like and problematic that, like I said, the authors, I don't have it right in front of me, but I think the paper was retracted maybe 5 years later, where the authors attempted to redate those sedimentary layers, like I said, just to put it closer to like where they believe that dinos and birds would have evolved. But yeah, a lot of these transitional forms, they'll suffer from what's called the grandfather paradox. For example, like if you looked at Tiktaalik, they found Tiktaalik where they thought it should be, but then years later they found tetrapod tracks that predated Tiktaalik 20 million, but by 20 million years, for example. I think it was in Poland they found these tracks. So I'm not sure if you're aware of the grandfather paradox, but yeah, take your time. If you want to address anything that I said, you're free to.
Oh, do you have that link to the paper with the bird tracks by any chance?
I do. Let me, I'll try and post it into the chat if you'd like to.
Awesome.
If there's anything you want to address while I do that, go ahead.
All right.
And
cool. All right. Okay. Thanks. So you mentioned about like how they're also like when looking at Tiktaalik and fish and then amphibious types of animals. You mentioned how there are also like there's submarines and then there's like amphibious types of vehicles that we make, and then obviously cars. The only thing is I see where you're coming from. It's just the difference is that those vehicles are designed by humans, whereas directly made by humans. Whereas Tiktaalik and these fish, they're organisms that reproduce, and over time, if there's isolation, you can get speciation, and so if you when you accept speciation, the only thing that will change, like from fish to something like Tiktaalik, is the amount of time. There has to be enough time for that to occur. So, yeah.
Right. No, and I appreciate that response. I think, to be honest with you, from my position, adding reproduction would just add complexity, and I think that would make the problem even that much more difficult for the evolutionists. But technically, our model and the predictions and the retrodictions that we would make are based on the fact that in the Bible it says we're created in the image of God. Therefore, reproduction, and I understand evolutionists will often say that, cars don't reproduce, but we're looking to the way we design things, since we were designed according to our model by God. Therefore, it comes naturally, which is the thing, right? It's not like we even do it on purpose, for example. So if we look at nested hierarchical patterns, I know evolutionists will look to these patterns that are found in anatomy, physiology, morphology, genetics, that seem to be consistent with descent with modification, right? But naturally, just by accident, we're not even doing it on purpose. Humans, we're designing things in these groups within groups patterns. So if you were to look at the types of vehicles and modes of transportation, for example, David, that we've created, they naturally form these groups within groups patterns. So real quick, a sedan has a tremendous amount of similarities with other sedans. Now we can bring in a semi-tractor trailer, for example. We would both agree, I assume, that a semi-tractor trailer is obviously much different than a sedan or a car, but these two modes of transportation are similar enough to each other that it sets them apart from airplanes. That's that groups within groups pattern that we see in the biological world. So if we were to look, and we would make this retrodiction, and we were to look at biological life, and then we were to compare it with the design world, and it didn't turn out that we built things like amphibious assault vehicles or built in homologous patterns or nested hierarchical patterns, then I understand what you're saying. There would be a point to be made, but it just so happens that we do. But plus, I think it also comes down to common sense too. And you can tell me if you agree with this or not. I think from both models, the design model and limited ancestry and universal common ancestry, that we should share more in anatomy, physiology, morphology, and genetics with a chimp than we would with an old world monkey, or than we would with a fish or a dog, for example, right? So I think it's kind of common sense, and that's why I always say, okay, it's kind of like saying, hey, listen, the sky's blue, therefore evolution's wrong. Well, you'd say, excuse me, like both models can explain that data, right? We got to look at that which is differentiating. And that's why I agree with you that it's genetics, it's genes that are inherited, right? I mean, we can start with like the chimp human similarity in DNA if you wanted to, and just see how close that is as well. But I do agree with you that it is in DNA that we can directly answer the question of ancestry. But I know I said a lot there. I hope some of it made sense to you. So you can take it away, David.
Yeah, sure. So yeah, again, like we have more genetic similarities with the great apes than we do with like a dog, or a frog or something like that. But since DNA is used to trace our relationships with other people around the world, the only thing is, like, I don't understand exactly where it break down when it stops becoming like a hereditary indicator and starts becoming like an indicator of design. I'm not exactly sure where it stops, determining relationships and starts indicating design.
Yep. No, I agree with that. If you're done speaking there, that's a good point. The question would be, and I believe there are a number of lines of evidence that can differentiate and draw a line in phylogeny. Before we get into that, let's take it one topic or line of evidence at a time. So if we look at human and chimps, we are, and I've read through the original paper where they came up with the number that they have, and what we're doing is we're comparing roughly 2.2 billion of our actual total letters, and the total amount of letters in the human and chimp genomes is 3 billion roughly, right? I mean, we pass on 3 billion from mom, 3 billion from dad, but they look at a particular type of DNA change when coming up with these numbers, SNPs, for example, single nucleotide polymorphisms. Now, there are whole chunks of DNA that are actually not found in either species, chimpanzees and humans. Now, if you include, David, the single letter differences, the chunk differences, and the big chunks that are missing on either case that I'm talking about, you only get about 88 or 89% similarity. Now, within that 88 or 89% similarity, I would agree with the original paper and with what I believe you probably assume the percent identity is as well. You can find between 95 and 98% identity within that 88. And there's a creation scientist, he's a geneticist named Dr. Jeffrey Tompkins. You've probably heard of him, but he's been making this point all along. It's basically, for the audience, if you include everything, you are upwards around 300 million DNA differences, and that's a lot. Now, real quick before you comment on that, I will say this, though. The point here is that we are a lot more different to the chimp than a lot of people make it out to be. But I would agree with you that we are still more similar to the chimp than we are to anything else in the biological world. So if we really are only 88% similar to chimpanzees, that means we're just going to be even less similar to a gorilla or an old world monkey or a dog or a fish down to a bacteria. So I'm not challenging the hierarchy that still exists, but the percent identity that evolutionists say we share with chimpanzees, I'm saying that is a little bit erroneous. Go ahead, David.
All right, cool. So you mentioned Jeffrey Tompkins. Oh, and I should have mentioned I'm definitely not a geneticist. I just know very, very basic stuff about genetics. So I just want to clarify that.
No problem. And I did want to say for later on after the discussion, if you want to look it over, I did post the paper from Nature. I posted it in the chat. I'm not saying you have to read it over now, but it is there. But yeah, take your time. Whatever you got to say to what I said, no interruptions. Just take your time, David.
Thank you. Yeah, awesome. So Jeffrey Tompkins, he mentioned about there was like only an 88% or something like that similarity as opposed to like a 96%.
So Jeffrey Tompkins agreed that we are 95 to 98% similar to the chimpanzee, but that's within that 88 to 89% similarity when we consider the chunk differences, the gaps, the copy number variations, the single letter differences. So within that 88, yes, we're 98% similar, but that's ignoring 12% of the actual genomic comparisons is the point. So it's more or less an agreement. Yeah, go ahead.
All right, cool. Okay, so this is pretty interesting. Yeah, I've never heard anything about like an 88% similarity. So the only thing is, like, how come this hasn't been like embraced by the overall scientific community, though? That's just like what I'm kind of puzzled about. So
Good question. Well, both creationists and evolutionists, anyways, would agree that humans and chimps are a lot more similar to each other than any other form of life. That hierarchy is still there. But even the evolutionists will acknowledge, you can see this in the original paper as well, that there are big chunk differences, there's single letter differences. Now, assuming that chimps and humans did share a common ancestry, well, obviously they have their explanations, for example. So since the divergence, let's say 6 million years, correct me if I'm wrong, from the split between humans and chimps, they would have been evolving separately. Therefore, there could be gene duplications, there could be deletions. So all of those differences, those big chunks, those gaps, copy number variation, size differences, the reason why they say they're ignoring them for the most part is because they believe those are the differences or areas in both our genetics that have changed and diverged since the initial divergence. Problem is, we don't assume that humans and apes have descended from a common ancestor. So we don't really need to invoke that explanation, but the evolutionists do. So from my understanding, they would agree with what I'm saying, but they have their reasoning, if that makes sense. Does that kind of make sense?
Sure, yeah. So, but also, like, it's kind of puzzling too, if they don't share a common ancestor, like, why do we find these mosaic fossils in, like, for example, the Australopithecines, like with these apes with very human-like characteristics, like their femurs, and their pelvis and stuff like that. Like, and why do we not find any modern humans in the same layer as these things?
Right. So that's a really, really good question. So when it comes to the so-called pre-humans, when it comes to the Australopithecines, Australopithecus afarensis, for example, or Africanus, according to our model, and I believe this is what the evidence would suggest as well, is that the so-called pre-humans that you would point to as our evolutionary ancestors, like Erectus, for example, or Homo floresiensis, the hobbits, right? Heidelbergensis, Neanderthals, Ergaster. I mean, we can go on and on, of course. The evidence seems to suggest, though, that they weren't actually pre-human, and they're better understood as human people groups, David, who underwent accelerated genetic degeneration due to severe and extended inbreeding. Now, why was this? According to our model, is this was all because of post-Babel migration and isolation. Now, with the Australopithecines, I do believe that they have some unique features. I agree with you there, David. Now, the Australopithecines, though, overall, they share most of their features with living apes today. They were only around like 3 or 4 feet tall max. And like I said, yeah, they did have some uniqueness in their morphology. I'm not going to disagree with you on that one. That just means I think the best conclusion regarding the Australopithecines is that these apes were nothing more than just an extinct ape genus, for example. And I do want to point out to you in the audience, I would have to pull it up, but I do have some an extensive amount of papers kind of supporting what I said about the rapid degeneration of these people groups. For example, there's a paper here that shows that Neanderthals, certain groups of Neanderthals, they were highly inbred and had a very high genetic load. The Homo floresiensis, the hobbits, for example, that were discovered on the island floors, I believe, they experienced island dwarfism. And what's fascinating too is on that island, there was also elephants that experienced island dwarfism as well. And I can show you that as well. But yeah, it shows that they were also highly inbred, and they, along with Naledi, Erectus, they suffered from reductive evolution. And what this reductive evolution leads to is reduced body size, reduced brain volume, and other various pathologies. So what's funny is all of those so-called features that evolutionists will point to and say, these are primitive. And I did have an extensive debate with Eric on this. It was like our first debate. So they'll point to those and say, okay, these are primitive. We're slowly evolving into modern man. But those are known to be due to this reductive evolution, right? All of these pathologies can be seen based on that accelerated degeneration. And that would make sense because migration patterns after Babel, a few people groups, unfortunately, whether it's the hobbits, Neanderthals, Erectus, Naledi, they get isolated, right? And then environmental changes, for example, if they're stuck on an island like the hobbits, it leads to rapid and accelerated genetic degeneration because they're inbreeding. Inbreeding is the exposing of the hidden reservoir of genetic mistakes. So I know I talked a little bit there, but I was just kind of, as quickly as I could, I wanted to explain how we fit those so-called pre-humans into our model. But there is supporting evidence, like I said. I mean, I've got a ton of papers going over this regarding the Neanderthals and the hobbits. So take your time. And what are your thoughts on all that, David?
Yeah, just like I want to clarify, what exactly do you mean by reductive evolution?
Okay, so good question. So reductive evolution for one is known in bacteria, for example. They'll lose genes short term for adaptive purposes, but what it leads to is shrinking functional genome sizes. Now, when it comes to these so-called primitive pre-human people groups, what it means is that they are accumulating so many recessive deleterious mutations that instead of experiencing, let's say, forward evolution, they're actually degenerating. They're devolving because those recessive mutations that are destructive and deleterious, they're coming to the forefront. Now, the reducing nature and all that is because the brain volumes are now reduced. The body size is reduced, as we see in a lot of these people groups that evolutionists point to. The brain volume, a number of pathologies. So that's what we mean by reductive evolution, because it's resulting in reduced, right? That's where the word reductive comes into play. Reduced, like I said, body sizes, brain volumes. Sorry, go ahead.
No, you're good, man.
Yeah, no, I was just saying a good one to go into on that would be, are you familiar with Homo naledi?
I can't remember exactly which one. That name sounds very familiar. Is that one of the more primitive ones, like habilis?
Yeah, I would say like Naledi, habilis, Australopithecus sediba. Like these are your stronger, I would say, cases of transitional forms between an ape-like ancestor to humans. Naledi seems to be one of the more, I guess, inferred ones or claimed ones. But that one, there's been some heavy research done on Naledi as well, and that there is evidence that they were also an isolated population of very tiny, kind of like the hobbits, genetically compromised human beings, and all the features that you see on Naledi. And Erica and I discussed this in one of our debates. They as well, Naledi had reduced body size, reduced brain volume. For the audience sake too, and your sake, what's interesting about the small brain sizes, David, is that under starvation conditions, okay, as these people groups would have experienced after Babel being isolated, after starving conditions over many generations, natural selection will actually favor reduced brain size, okay? Because the brain, as you know, is the most energy-consuming organ of the body. And we can see this observed today in mammals that are living in isolation, for example. So you'll see, like, you can look at Bill Ludlow or Erica's presentation where they will line up like the brains of like Australopithecus up to like habilis, Naledi, up to Erectus, up to Neanderthal, like from small to large. But the fact is, that's not what we see. We're seeing, as I said earlier, reductive evolution due to the isolation, the inbreeding, and it's just a basic scientific fact that, yeah, natural selection will favor a reduced brain size. Now, this isn't leading to forward evolution. It's kind of like the bacteria, and you can even just Google, right? Bacteria, reductive evolution. For adaptive purposes, especially in the lab, they will just lose genes. They're throwing out genes for short-term adaptive ppurposes. It's almost like if I wanted to get better gas mileage in my car just for the time being, so I'm like, oh, what can I do? I'll reduce weight, for example, start throwing off the doors, throwing off the door handles, whatever, just to reduce weight, just to get better gas mileage for that moment. But overall, it's not beneficial. It's destructive, right? So that's what we're seeing in these so-called pre-humans. So we would say these were modern people groups. They they migrated, became isolated after Babel, and they, I don't want to say devolved, but in a way, they became genetically compromised into what evolutionists now say Homo erectus, heidelbergensis, naledi, habilis, whatever, you name it. Well, the evolutionist says, no, these were the people groups that, well, a lot of them lived at the same time anyways, but they would have been the precursors to modern people. So it's kind of like it's opposite views of these so-called people groups. But like I said a lot there, so just take as much time as you need there.
Cool. So you mentioned like all the different pre-humans or proto-humans or whatever you'd like to call them, like the Australopithecines and like Homo erectus and so forth. They're all dated at different time periods though. So like, the Australopithecines, they appeared before, Homo habilis and Homo erectus. They're not just like lining them up based on like brain size. There's also like a time constraint as well. So
right, that's a good point. But even experts in the field that are not young Earth creationists, they're describing these various Homo variants, right? Like the Neanderthals, Denisovans, Erectus, Hobbit, Naledi, we can go on and on, as actually being part of a large interbreeding metapopulation. And Erica, who's, I think she's studying anthropology, she agreed with this. And we both compared it to kind of like if you're a Lord of the Rings fan, you've got Middle Earth, and you've got all these, you got the dwarves, you've got the hobbits, you've got the elves, you've got the orcs, they're all living in the same world at the same time. So now even experts are recognizing that that all of these so-called pre-humans, they were simply part of a large interbreeding, quote unquote, metapopulation. So they coexisted, interacted, and interbred. So what you said about living at all different times is actually not correct. I think they used to think that, but the data and the field is changing rapidly.
Right. So like Homo sapiens, we did coexist with Homo erectus and with the Neanderthals. But like, for example, we didn't coexist with like the like Afarensis or I'm trying to see about we didn't coexist with Homo habilis. I'm sorry?
Are you saying like we didn't coexist with like you'd agree that these Homo variants
not all
existed, but okay,
right,
which one?
With Homo erectus, but not with Homo habilis.
Okay, so Homo habilis. Actually, first let's discuss the Australopithecines, I guess. So I've already went over like what how we would explain Lucy, for example, but with some latest findings, I discussed this with Erica as well. They found modern looking human footprints on an island in Crete that actually predate or are dated around the same time as the Australopithecines. They found modern human-looking footprints in Mary Leakey found it as well. Like these anomalies have been discovered, and they've oftentimes found Homo-looking bones in the same fossil layers as these Australopithecines, for example. But then they always come up because their basic assumption is, humans didn't live with the Australopithecines. So now they come up with different interpretations of the data. For example, those footprints in Crete, Erica has an extensive video on it where they're trying so hard to look at the details and say, hey, maybe this was, well, for one, maybe these weren't footprints, but that seems like kind of a ridiculous claim. Or maybe they were like a variant of gorilla species or something like that. But my point is that claim you made kind of comes down to, for one, the assumption that those layers are a reflection of deep time evolution, and two, assuming that the Homo groups didn't actually coexist with the Australopithecines. So anytime they see data that contradicts that, it's kind of like circular reasoning, like, oh, these footprints look like modern human footprints, but we know that humans didn't live with the Australopithecines. So now we got to do further research to show why these really aren't modern human footprints. But yeah, go ahead, David.
All right, thanks. So, like, in order,
I want you to
in
know
terms of,
that.
like,
When you call me on the phone,
like
you saying all this,
saying
and you
that
get on
these
the live
layers are
stream,
different
and you
ages,
doing this-
we date the layer.
You
That's
should
why
be ashamed
we
of
assign
yourself.
different
Is that
ages
black?
to them.
Is
We
that
date them
black?
radiometrically.
Is
That's
that
how we
black?
date the ages.
You's a bitch. I called you.
Right.
That's
What?
your
damn.
mouth.
Like
You's
what?
a bitch.
Oh, you gotta call
I'm
it
sure
the
you've
word.
heard our explanation that
I like it.
these
You wanna
dating
be
methods,
hood,
and I
nigga?
definitely don't want
You
to go
wanna be
too
hood?
far off topic
You
because
wanna
we
bring
definitely have a lot to
shit to
discuss
me, and you call me
on
with-
genetics,
You a
ancestry.
bastard?
But actually, I kind of like that we've gone
I
into this
never
field because
Look
we
at
have
his
a lot
face.
of
Look at his
time
face.
Yeah.
here, detailed discussion on it. But
said
the fossil
what I
record
said-
requires
what
so
I
much
said was-
inference.
That's what I
There's
said.
no
I feel at home
genetics that
now.
we can actually use. There's no genome sequencing, for example,
Fact.
because you're asking
All right.
how do we draw the line
So
in
that was it.
phylogeny,
That was
right?
the-
And I'll get to that real quick because you mentioned the dating methods. Well, generally, like I debated Conspiracy Cats, he'll admit that all these dating methods, they require assumptions, okay? But he'll point to isochron dating as the number one best way to date rocks and date layers without the assumptions, right? But what the evolutionist doesn't understand is that even isochron dating relies on at least one unknowable assumption. So the other dating methods, they assume initial conditions, constancy of rate, a closed system, for example. But isochron dating even requires an assumed history. And what I mean by that is isochron dating assumes that there was no global flood that accelerated nuclear decay. So that isochron dating at least has that assumption to it. But go ahead.
All right, cool. So you talked about like assuming like initial conditions or starting conditions. The thing is, like, they count the ratio of parent isotopes to daughter isotopes.
Right.
So like when like C14, or I forgot what exactly it is. I believe it's C14 decays into some other type of
yeah, like carbon. Yeah, carbon they wouldn't want to use to date anything over like 50,000 to 100,000 years.
Exactly.
So yeah, they would be using like potassium argon dating or
You're
these.
absolutely right.
Right. Yeah.
Thank you. Yeah, thank you for that, by the way.
No problem.
Yeah. So like, you compared like the ratio of potassium to argon. So it's the starting conditions. It doesn't really. It's not necessary to know what the starting conditions were. So you just have to compare ratios. So.
Right. That's a good point you made. Unfortunately, though, for example, argon, okay, is present in rocks of known young age, meaning this assumption of zero argon in rocks of unknown age is actually an unreliable and kind of a ridiculous assumption. Because my question would be, why in the world would we trust these dating methods if many of them can't even give accurate dates for rocks of known age? But now we're supposed to trust these dating methods of rocks of unknown age. What that means is they're taking rocks of known age and they're measuring the ratios like you're talking about of isotope decay, and these rocks are coming back as millions and millions of years old, even though they were just formed due to a volcanic eruption or something. But my biggest point would be about the assumed history that obviously, according to our model, and this would be going into a global flood debate, so we don't have to spend too much time on it, though I am interested in your thoughts. If radioisotope decay was accelerated, say during the Genesis flood, as we would propose, then the radioisotope decay clocks that we're talking about here could never actually be relied upon when they date rocks of millions and billions of years old. And the fact is, we actually have several independent lines of evidence that show that accelerated nuclear decay did occur in the past. So yeah, even if some of these dates do come back as millions of years old, with evidence for rapid nuclear accelerated decay, it's more than plausible that the rocks are really only a few thousand years old since the initial flood event. But go ahead.
So you talk about accelerated nuclear decay. So like, are you talking about like, do you think that like the decay rate of certain isotopes was somehow quicker in the past or something like that? Or Right.
Good question. So because of the global flood, we looked at catastrophic plate tectonic movement during the flood, which would produce a lot of heat. There would be subduction, friction, runaway subduction, for example. There would be a lot of activity going on in the mantle and in the rocks as well that would speed up these decay rates. And now there's been a group of scientists called the RATE Project. They've undertaken a research project to look for these lines of evidence, and they've I'm not sure if you're aware, but byproducts of radioactive decay include helium, fission tracks, as well as radio halos. There's other things too, but I don't want to spend too much time on it. But that has been seen in the rocks. So that's actually observable evidence in the rocks that rapid nuclear decay has occurred in the past. So yeah, we would say because of the flood and because of the excess heat that would be, I guess, created because of the flood event, that would speed up these rates, accelerated nuclear decay. Go ahead.
All right. Yeah, that's pretty interesting because from what I've heard, I've only heard that like the decay rate of like, potassium turning into argon. I've heard that that's, like a constant, kind of similar to like the speed of light, for example. So I'll have to look that up to see what you're talking about, like how like tectonic plates shifting like may have had an influence on the decay rate.
Yeah, I can show you some. Like you're right. Yeah, heat and pressure, for example, and like the volcanic activity will help speed up the decay rate. I mean, even today, heat and pressure can change the decay rate, but not that significant. But that's why the flood, which is a sudden, unrepeatable event. And I like the way Walt Brown puts it. He said this global flood, whose waters erupted from interconnected worldwide subterranean chambers, had an energy release exceeding the explosion of 300 trillion hydrogen bombs. You can find that in his book in the beginning. So like the things that I said, the rapid movement of the plates, meters per second movement of the plates, runaway subduction, friction, that can wreak enough havoc to accelerate nuclear decay in the rocks. Now, generally, a lot of, like, if you debate Gutsick, Gibbon, Erica, for example, or Aaron Rao, they may concede that point and say, okay, fine. But that would produce so much heat that would boil the oceans, for example. So they may go into a secondary argument and say, let's just say that the fission tracks, the helium, the radio halos you're looking at, let's just say, okay, there was some type of event in the past that has sped up nuclear decay. How do you explain the fact that there's going to be so much heat release that it's going to boil the oceans and kill Noah and whatnot? That's usually the secondary argument. If you're not too aware or you haven't read much on this, it's not like we have to go into that. But this is a discussion that's taking place within creation science and also between creation scientists and evolutionists. Go ahead, David.
All right, cool. Thanks. So like you mentioned creation science. How come this isn't just like regarded as just like science altogether? How come like it's divided into like, evolution and create? How come like what you're talking about isn't like just like described as like science in general, if you know what I mean?
Right. And that's a good question. I'm just kind of saying creation science just to make that distinction more so for yourself or the audience that, hey, listen, these scientists are looking at hypotheses. They're looking at predictions that can flow from those hypotheses to confirm whether or not the young Earth creation model has some, validity to it. So that's just kind of making the distinction. But yeah, it is science, and that's why, like, your question regarding phylogeny and where can we draw the line in ancestry, that's a good question, because we've made testable predictions based on our model of created heterozygosity, which is interesting because it suggests that Adam and Eve were front-loaded with these pre-existing functional DNA differences, and this would actually apply universally across species. So what's interesting is if a scientist showed up at the creation event and they started looking at anatomy, morphology, physiology, or they started doing genomic analyses, already right off the bat, they're going to see nested hierarchical patterns in DNA, in anatomy, in morphology. Now, a direct prediction of this hypothesis is mutation rates, DNA function, and speciation rates. So my question to you, as a way to kind of, you can never break the nested hierarchy because it exists. I find people like Aaron Roth, they think that God just created human beings equally distant in DNA, morphology, and anatomy to all types of life, as in like humans are equally distant to chimps as they are to fish. I mean, that's just ridiculous. Obviously, if
Right.
God used DNA, you know, then it's just on both models, it should be common sense. We're going to share more with the chimp than we do with the fish. That's why we got to look at differentiating evidence. So are you, one line that I can go over, and instead of just throwing a bunch at you, we can just go through one. Like, I would point to molecular clocks, but before we do that, and like you said, you're not a geneticist, we don't have to go into
Right.
too
Yeah,
much.
Don't know.
So I'm not,
definitely.
it's just a friendly discussion, so I'm not here to trap you or anything, but something like orphan genes, which isn't too highly technical. Are you familiar with orphan genes there?
No.
Okay. Okay, so, well, if you think about it, just the name orphan, like what would, if it's orphan and then gene, like what would that mean to you, do you think?
Yeah, like an orphanage, like somebody whose parents, had to like adopt them out, not to a specific person. I believe. I hope I'm saying that right.
Yeah, no, that's good. That's good. So there's these orphan genes that, here's the problem for the evolutionary model regarding animal origins and orphan genes, is that these orphan genes, they're novel DNA sequences, and the unique traits that they are associated with, because they're functional DNA elements, they appear suddenly and fully functional without any trace of evolutionary ancestry. So like you said it absolutely correct. No evidence of parents. You know what I mean? Like, these are lineage specific DNA elements that are unique to, say, the humans. You can find ones that are unique to the chimps, to the dogs, to all forms of life. You know what I mean? So like, my question would be, and I've debated biologists on this, asked Erica this question too. I said, how does the evolutionary process explain the origin of these novel but yet functional DNA sequences that are specific to so many different kinds of life, right? Because you want me to draw a line in the phylogeny. Well, I like to look at orphan genes as a way to say, look, at humans have these orphan genes that are highly functional DNA elements that, say, the dogs don't have, or the whales don't have, or some of the great apes don't have. I can use that to build a tree and to do a genomic analysis. And the key is that these would have to be taxonomically restricted, but they'd also have to be essential to the genome in order to conclude something like this. Yeah, what are your thoughts? What do you think about that?
So like, regarding orphan genes, are you talking about like specific genes that are unique to certain organisms?
Right. So not just unique though, not just like junk areas or anything like that, like unique, but also highly functional. You know what I mean? It's almost, because according to our model, so the whole reasoning, David, just to kind of break it down easily and for the audience, the whole reasoning for the existence of these orphan genes is based on the fact that, like I said, they appear suddenly. Boom, there they are, with no evidence at all for ancestry, and they have no similar counterparts in other genomes.
Well, can you give me an example, like, of an orphan gene that's highly functional, or just to get an idea?
Yeah, I mean, I can send you paper after paper. Technically, all of these orphan genes, for the most part, I mean, obviously, we know so little of the DNA language, so we're still doing genomic tests. But the ones that they've sequenced, the ones that they've tested, they are known. Even the evolutionists will say that they are functional. Now,
what do they do though? That's what I'm confused. Like, what exactly do they do?
Okay, that's a good question. So they can be used for immune responses. They can be used, like, they'll code for a functional RNA messenger. They code for proteins, for example. So there's a number of different types of functions specific to these orphan genes. I'd have to look at the paper to like really go through the specifics, but the thing is, if you look at these papers too, they will often say like, you know, where did these orphan genes come from? How did they come about? And they will look to what's called de novo gene synthesis. So, and the reason why this, like I said, because these orphan genes, they're fully functional gene sequences. Like I said, they code for both a functional messenger RNA and a protein. They lack similar counterparts in other organisms, like I said, but that's why they have to fit it into their evolutionary story. So de novo gene synthesis, what it is that for whatever reason, mutation, environmentally related, a non-coding region of our genome suddenly popped up and became functional as as a protein, for example. So that's their explanation. Because I'm just here to have a discussion, not here to trap you. So that's what they would say. Do you think that would be a good explanation, you know, that they're coming up with, even though, now remember, they're inferring this though, right? They're just saying, okay, this must have come about from the non-coding regions. But according to our model, this is exactly what we would expect, given the uniqueness associated with, say, the created kinds that we look to in Genesis to now make predictions, retrodictions. Yeah, go ahead, David, take your time.
Yeah, honestly, I don't really know enough about that specifically to say either way. I'd have to definitely do some homework on that, if you know what I mean.
Of course. And that's why, you know, I oftentimes, if I'm having discussions, I also like to explain the other side as well, right? De novo gene synthesis. Like, for example, I oftentimes like to go to because we can look at lineage-specific retrotransposons. So we can look at ERVs, ALUs. I mean, there's a number of classes of retrotransposons that evolutionists will often point to as the number one best evidence for evolution. Like Conspiracy Cats, if you're familiar with him, he brought that up with Hovind. He believed that that's the number one evidence for evolution. But now these ERVs, we know that they act as DNA regulatory elements, as long non-coding RNAs. They are involved in the immune system. I mean, I can just go on and on about their functional roles in determining cell types, for example. But these are all direct predictions and expectations based on that model I was talking about, of created heterozygosity, where these functional DNA differences were front-loaded into Adam and Eve and the species. So we should see all these functional roles, not a genome of junk, as the evolutionists would suggest. Are you familiar with like the ERVs and the ALUs and those DNA elements, David?
No, I just have like a very basic understanding.
Well, that's okay. That's okay. Like, what would you say is, like at the top of your head, what do you believe is just the number one best evidence for universal common answers? Like, I know you went over a number of things. Is there anything you feel like we haven't discussed in enough detail? Like maybe the geographical distribution of organism marsupials in Australia, for example. Like, what do you believe is the number one best evidence? Go ahead.
Oh, well, I still think the number one best evidence is probably genetics for universal common ancestry. Given that it's used to test heredity. Regarding testing our relationship with other people. They use the exact same test to determine our relationship with the great apes, or with pretty much any organism. But regarding the geographical distribution of organisms, like in Australia, like with the mammals there having pouches, that seems to make sense to me about evolution, regarding how it's been isolated for the past, I don't know how exactly long, but it would make sense why they would all mostly be marsupials, with a few exceptions like bats and a few rodents. But that would make sense to me. So yeah,
that's and that's a good line of evidence. That's some good questions that you'd have there. I'd say just before I answer that, I would just say regarding the genetic data, I believe that, and we can even save like a more detailed discussion on some of these lines of evidence for a future date. But molecular clocks, which I suggested before about the for example, the mitochondrial DNA, the Y chromosome, which goes back just 6,000 years to Adam and Eve. Of course, we can see, like you talk about geographical distribution. we can see the geographical distribution. I think, actually, did you have a comment on the mitochondrial Eve and Y chromosome atom? You said
From what
something
I've
there.
heard, I'm definitely not an expert by no means on this, but based on what I've heard that the Y chromosome atom goes back about 50,000 years, something like that. I probably have that number wrong, but based on what I've heard, something like that. I don't recall anything like 6,000 years. Do you have like a paper, or do you have like a source for the 6,000 year?
Yeah, I do. So instead of screen sharing and go through all of them, so even like secular pedigree-based studies on the mitochondrial DNA, not even from creationists, they just use the empirical method, right? So they're not calibrating it with the fossil record or phylogeny-based assumptions. They're just looking at how quickly does the DNA change from generation to generation. So mitochondrial DNA clocks are actually generally pretty fast. So they can take this observed mutation rate and they can just based on the empirical method, they can trace the origin back. Like there's only a few mitochondrial DNA differences that separate us and the mitochondrial Eve sequence. And that only goes back about 6,000 years. Now, what you said is true. The evolutionists will say mitochondrial Eve goes back a couple 100,000 years ago. But remember, that's not based on the observed mutation rate. What they do, David, is they have to calibrate it now to the fossil record and to the phylogeny-based assumption. So what they do, and you can watch my third debate, I believe it is, with Erica. We talk about this for about 30 minutes. They invoke mutation rates that are like 10 to 20 times slower than actually is observed, because they need to now stretch that out 250,000 years. So what's funny is the creationists are the ones that are using the empirical method and consistent rates for the most part. Now, somebody like Erica, or I'm not sure if you're familiar with like Herman Mays, Dr. Herman Mays, yeah. He's a very intelligent evolutionary biologist. He will admit to this, and he will admit that we have to assume the fossil record. But what's funny is that's circular, because as a young Earth creationist, that's exactly what we're questioning, is the deep time and the fossil record. So if we have observed mitochondrial mutation rates that only go back 6,000 years based on a consistent mutation rate, there's absolutely no reason for us to now calibrate that with the point in question, which is the fossil record. But here's the main point. Evolutionists can invoke slower mutation rates in the past that are 10 to 20 times slower than observed, just to calibrate it with the fossil record. But for that to be a scientific explanation, David, that would have to lead to testable predictions. Now, here's where they will admit that they don't have testable predictions, because there's too many factors at play. But you might be aware of this. Dr. Nathaniel Jensen, a Harvard graduate, he's a young Earth creation scientist. He has made predictions. He said, okay, let's take this consistent mutation rate in non-African people groups. Now, let me show you how confident I am with the consistency of the mitochondrial DNA clock. I'm going to make specific predictions, future novel predictions, on African people groups where their mutation rates have not yet been measured. So for example, a mutation rate in the Khoisan peoples, they're an African tribe, they haven't been measured yet. So he actually made a direct prediction. He pointed at them and said, this is what I predict they will change based on the known mutation rate. So we, as creation scientists, we have testable predictions in print to go with the observable data. The evolutionists, yes, they have their explanation about slower mutation rates in the past, but here's the important distinction. They don't have the testable predictions to go along with that explanation. That means it's post hoc, ad hoc reasoning. Go ahead, David.
Okay, wow. Yeah, that's quite a lot.
It's a mouthful. No, I know. It's it's a mouthful.
Yeah.
If you want to just like take some time over the next few weeks and study that, and we can discuss it more, I can go into the kangaroos. Or, I mean, you're a guest here. You can take as much time as you need and give me your thoughts on that. It's laid back, easy going. So, like, how would you want to proceed, David?
Yeah, thanks so much. Yeah, you're awesome. Yeah, I have quite a bit of stuff to look over. So before I can address this kind of stuff. So
no problem. I mean, it's just a respectful back and forth dialogue. We're here to learn from each other. I oftentimes, like, because Some creationists, they may use out of date arguments. Not saying it's bad, but science, especially in the field of genetics, it has improved and advanced so much that if like creationists, if they're not up to date on the literature, both in the secular journals and the creation journals, and I know evolutionists, they scoff at the creation journals, but those journals actually will say, evolutionists, they can write in the creation journners and submit a paper and say, hey, I disagree with, like, let's say the mitochondrial DNA mutation rates or something. And Nathaniel Jensen, Harvard graduate, he will respond. And that's happened before too. So I would just say if they have a problem with the AIG journal, for example, then they can just submit their paper refuting it. But with the kangaroos, go ahead, David. Yeah, go ahead.
Well, that's another thing, is to still confused as to why it's divided up into a creation journal and an evolution. Why not just just a journal, not just one journal? If you know what I mean.
I do, and that's a good question. And here's why. So I'm staring right now at. You can go look at my opening statement with RJ Downard. I showed all the papers. I'm staring at 5 or 6 secular papers on the mitochondrial DNA mutation rate, where the pedigree-based study itself takes the mutation rate back 6,000 years. Okay, so that's good. That's good for us. But then they go through the paper, especially in the discussion portion, and now they're figuring out ways to calibrate it with their own date. So remember, those secular journals, they always have to assume evolution. Now, don't get me wrong, they can question the details. You see these evolution scientists, they're always questioning the details, but they never question the bigger picture. They never question the actual theory itself. So creation scientists, no matter how, Dr. Nathaniel Jensen, he publishes, a mitochondrial DNA mutation rate paper in a secular journal. It's not going to get published because he's not assuming deep time evolution. So they have to have their own journals. Well, but yeah, go ahead.
Well, I don't understand why there would be some conspiracy to filter out any viewpoint. I just don't see the motivation for a conspiracy like that, though.
I wouldn't say it's a conspiracy, because there still are intelligent design advocates that do get papers published in secular journals. Dr. John Sanford, he wrote the book on genetic entropy. He's got paper after paper that are published in the secular journals. He has actually just recently spoken. He's a young earth creation scientist. He's a geneticist. He invented the gene gun, for example. He spoke at the NIH, the National Institute for Health. So it's not that they can't get published, for example. Are you familiar with John Sanford and genetic entropy and mutation accumulation and this premise?
Genetic entropy. What do you mean? What do you mean by genetic entropy exactly?
So good question. So it's the fact that, we've gone over genetically why our genome is more and more data, more and more accumulating papers is suggesting that the genome is near fully functional. There's a project called the ENCODE project. It's a secular project. They reveal that over 80% of the genome is actively transcribed in RNA suggesting function. Now that's biochemical, so we don't know what every single gene, every single RNA is doing. But we know that there's activity there. And why would the cell transcribe all this junk? It would just be a waste of resources and energy. So we know that there's activity there now. Well, we're actually, creation scientists are making predictions that, hey, you knock out these genes, for example, and they're going to have this function or this function. So there's a lot of predictions coming from our side on DNA function. But the evolutionists assume the majority is junk, so they can't really make those predictions, unfortunately. But genetic entropy would suggest, okay, so our nuclear, we have our somatic cells, germ cell lines. Reproductively, we pass on about 100 new mutations per person per generation. Okay, so those accumulate. But the thing is, natural selection only acts upon the most detrimental of mutations. Boom, gets rid of it, right? And acts upon the best beneficial mutations. So let's say sickle cell anemia, melanoprotein, whatever, you can name it. But it doesn't do anything against the nearly neutral mutations because they're invisible to selection, because those mutations, David, they only have a very subtle effect on phenotype and genotype. So genetic entropy would suggest, okay, kind of like a single spelling mistake in a book the size of an encyclopedia. One single spelling mistake on its own, it's inconsequential to the message, but the buildup and the accumulation of them over time will degrade the message and degrade the book. And that's what we see in our genetics. That's why man appears to be devolving. That's why there's like the genetic database of diseases is just growing every single year. It's entropic degeneration on every level. So the question for somebody who, let's say, disagrees with genetic entropy and the fact that man is presently devolving, the question would be, and the question I would have for you if you disagree with that is, what type of selection could rid the population of so many mutations that are just pouring into our genetics that natural selection can't even see? They're essentially invisible. Yeah, go ahead. What are your thoughts on that?
So when you say we're devolving, are you like saying that like we're becoming more susceptible like to diseases or something like that? Or
yeah, good question. I'm just saying that we are accumulating. According to our model, God created us perfect genomes. He created us not to reproduce by cloning.
What do you mean by that? Like,
right. So we know that mutations are typographical errors in a text. You know, we accumulate, each gene has a different mutation rate. Mitochondrial DNA is going to mutate differently than the nuclear DNA, but the ones that are passed on generally in nuclear DNA in our germ cell lines is about 100 new mutations per person per generation. So mutations we now know are generally destructive. They're either nearly neutral or detrimental. And even the ones that are beneficial, like sickle cell anemia, they're still reductive in nature. There's a benefit and an adaptation to be had, but it's typically due to like a broken gene, broken protein. So genetic entropy just says, hey, listen, things are breaking down. Species are going extinct. Genetic mistakes are accumulating. If you take, David, according to our model, it makes sense, fits perfectly. We can take this point right now, this point of genetic entropy, this point of accumulating mutations, we can take that back in time to what's called a point of least accumulating mutations, where there is no mutations, no genetic mistakes, just those pre-existing functional created differences by God. That is what we mean by a perfect genome, free of genetic mistakes.
So like regarding free of any mistake, so what exactly would like a perfect genome, like what exactly would that be like at a face level, if or at like a that doesn't even make sense what I just asked.
That's okay.
Yeah, but like what exactly would like a person, what would that person look like with a perfect genome or a perfect person?
A good question. So there's a number of ways we can go back to the original created genome, and we're doing it through, like, I'm not sure if you're familiar with, I know you're familiar with human chromosomes, but are you familiar with the fact that they're made up of relatively large linkage blocks?
A linkage block?
Right. It would just be like, because these linkage blocks would consist of nucleotides. It can be thousands of nucleotides within the chromosomes. Mm-hmm. And when we analyze these original text strings, it looks like, especially in the human genome, that because they exist, so every generation we experience recombination, okay? Mm-hmm. And that's why we look different. You know, me and my wife would produce our daughter, and recombination would take place, kind of like shuffling a deck of cards. There's something called gene conversion as well. So over time, these processes, these genetic processes, they scramble up the chromosomes. They scramble up the linkage blocks. So if deep time evolution was true, let's say we evolved from Homo erectus 200,000 years ago somewhere in Africa. And then before that, Homo erectus was evolving for millions of years. And before that, you have the Australopithecines. Well, that amount of time should scramble, especially if recombination and gene conversion are happening nearly every single generation. It should scramble our genome to randomness. There should be almost no linkage blocks seen. But the fact that we see so many of these linkage blocks, you can see this in what's called the HapMap data, tells us that our genome is young. So what's fascinating is because these linkage blocks exist, creation scientists like Robert Carter, John Sanford, they are tracing these blocks right back to the original blocks, the original text strings. And that would technically be an answer to your question is, what did this original perfect created genome look like? Because remember, if these DNA differences were front loaded into Adam and Eve at creation, well, it's funny because opponents of Adam and Eve, and I just did a response to Godless Engineer, they'll say, hey, listen, it's impossible. Adam and Eve couldn't have existed. Biologos will say this, that there's just too much diversity in just 6,000 years from two people, Adam and Eve. Well, for one, that assumes that all diversity is the result of mutation. All those nuclear DNA differences, evolutionists say, were due to deep time evolution. But we're saying that no. Most of these DNA differences were front loaded into Adam and Eve because God said, be fruitful and multiply. Did he mean for that to be carried out through cloning? It wouldn't make any sense for God to not front load Adam and Eve with DNA differences. So we can actually now look at the DNA differences around the globe. We can look at those alleles that are geographically specific and more rare. So we can say, okay, those are obviously mutations after Babel because they're geographically specific. But the ones that are more so common and not disease causing and functional and important, those are the ones that we can safely say, okay, those are the ones that were initially created at creation. So just take away all those mutations, those rare alleles that we find. I think there's about 65 million maybe worldwide. I mean, they're still doing genomic testings and analyses, but take those away, take the common variants, because you and I, we've got about 3 to 4 million places, more or less, in our that we got from our mother and our father that actually differs from each other within our own genome. So take the ones around the world, throw that into Adam and Eve, and boom. So I know that a lot of this is technical, but your question was kind of a tough question to answer. But
right.
Short answer,
you've done an awesome job.
We're working on it. You know.
Yeah, you've done an awesome job answering it. So yeah, thank you so much for being so darn good doing that.
Well, I want to say this, praise, and you've been awesome too, respectful. Because you're the guest, I want to just give you uninterrupted time right now before we go into. Well, it's probably been an hour and a half discussion, so we can, if there's a couple questions, or we just go right into the after show after. But uninterrupted time, just to kind of give your thoughts as to how anything you feel like I said that it wasn't addressed that you want to address, or just what are your thoughts about the discussion and everything that's been said, and do you still hold to the same lines of evidence that you believe are the best evidence for universal common ancestry? I'm just going to mute myself and you take your time.
All right, cool. Thanks. So yeah, you've done an awesome job, like addressing especially the genetic aspects of it. I'm not exactly sure if it's exactly true. I'll have to look it up myself. I have quite a bit of homework to do, so yeah. But yeah, you've done an awesome job explaining that in great detail. And also, the only thing I guess we didn't really address was like the marsupials in Australia, or like
right.
their geographical distribution.
You're right. That is the only
thing we didn't address. So yeah.
Okay. I'll give you. I think you're so respectful and fun and easygoing to talk to. You deserve an answer. So, according to our model, okay. So I'll try and explain it the best I can in a short way. So the ark landed in the country Turkey, for example. Then the animal kind began to spread out. Now. Kangaroos, for example, they're less aggressive than some of the carnivores. So they would be driven further and further away from where the ark landed, because we're looking at migration patterns after the flood. Why don't we find too many kangaroo fossils from Turkey to Australia? And, yeah, I think we do find a select few, but animals die by what's that?
Did we ever find any like kangaroo or koala fossils in Asia? Or
I don't think so, and that's why I'm saying that from Turkey because it would have been after the flood. So overall today, I mean, animals die by the bazillions, and they don't just turn to fossils. So yeah, catastrophic events and rapid burial, for example, can fossilize an animal, but after the flood, during those migration patterns, would they have even fossilized? It's tough to say. Many of these animals, I would say, that are migrating to all parts of the earth and that we see are now geographically specific, they wouldn't necessarily have fossilized. But how did they get to Australia? How did they become geographically specific. Well, if the ice caps were larger, as we'd say, according to our model, and the oceans were much lower, even by, say, and I explained this to RJ Downard in our last debate, but say even by 300 or 400 feet, what's fascinating is nearly everything, David, becomes connected. So in a world such as this, migration is simple. There would be land bridges literally 1,000 miles wide. Now here's the kicker. When the ice caps melt back and the water levels begin to rise, your kangaroos, your marsupials, they become trapped and isolated in places like Australia. And now their speciation process begins. And now you got all the different kangaroo species that came from the original ones off the ark that were already isolated in, say, Australia, for example. Right. So I think that can also be explained. To be honest with you, I think a lot of that, like the land bridges and stuff, that's how the evolutionists also explain a lot of the geographical distribution. Ours just occurred roughly 4,000 years ago versus millions of years ago. But I'll let you respond to all of that, I guess, in what this could be as your closing. So you just take your time. If you disagree with that, tell me why, and yeah, just take your time.
Cool. Yes, that's awesome. So like also, when looking for fossils, like we do find, like, kangaroo fossils in, Australia, of course, but we don't like find any trace of them in Asia either. So, I mean, they would have had to have been in Asia for at least some time. And also, when looking at other animals in Australia, that like you would probably think that like that koalas are a different kind than like kangaroos. I would think, right?
Yeah, well, I mean, I guess my argument to that would be the fact that nearly all the fossils in the world that we see today are due to rapid burial. So if this was after the flood, many of the animals, including the kangaroos, why we don't find them, say, fossilized in places like Asia during their migration patterns from Turkey to Australia is because those catastrophic events wouldn't have been taking place during that time to even fossil. I mean, how many animals die today? Millions. How many fossilized? None for the most part, right? So it takes very special conditions to fossilize an animal. So that's the best answer I have. I wasn't there, and that would be the best answer I have. I think it fits nicely, but yeah, go ahead.
What about like also for like sloths? Like since those are only found in the Americas, how come like we don't find like sloths in like Siberia? They would have had to have like migrated from Turkey to across over to Alaska and then, down into, Central and South America. How come we don't like find any sloth fossils in Siberia?
Well, I mean, I think it would be the same thing as the kangaroo. Like, why would we expect to find sloth fossils or kangaroo fossils? Because they literally just get off the ark and right away they start migrating for a number of reasons. I touched on a couple, but they wouldn't have been in any of these areas for long at all. And fossilization, as I said, is extremely rare.
That long though.
Well,
like, some of them stayed in in Asia or.
Yeah, well, I think because of the post-flood effects and the ice caps that are getting larger, and the aggressive carnivores are driving a lot of these marsupials, the sloths as well, further and further from where the ark landed. And the fact that fossilization takes very special conditions to begin with, it's not like they're just. I'm not sure if they stayed in an area for too long, but I think your question also assumes the deep time evolutionary history of migration patterns. This would have all happened within probably 1,000 years or so after the flood, and then the post-flood ice age, of course, would have been enough to, like, the ice caps would have melted back, water levels begin to rise, and now all your geographically specific animals are now isolated in the areas that you're talking about, the sloths, the marsupials, for example, those that are in Asia. Yeah, go ahead. Cool.
Yeah, that's awesome. I'm still a little skeptical about the speciation rates. I'm still a little skeptical about there being enough time for that to happen after the flood. And also I'm still skeptical about there being enough room even for all the different kinds that were on the ark. About there being enough room and food and water and all that stuff. So,
Yeah, no, and I've had entire debates on that, so that's even just opening up another
Right,
can
that's a different.
right. And I'll just say in 2 sentences, that just goes back to that idea of pre-existing genetic diversity. So if you've got like the cat kind, the bear kind, the dog kind, you name it, if they're on the ark and they've already been front loaded with these pre-existing functional DNA differences, all it takes is processes after they get off the ark, for example, recombination gene conversion, especially if those differences are already built in and doesn't require deep time mutation accumulation to explain change. Now you just look at migration, isolation, shifts from what we would say is heterozygosity to homozygosity, and boom, you got new species. And I debated a zoologist on this on Modern Day Debate, and we went into specific examples: lizards, bird species, snake
Mark
species. Not Mark, it was Adam Heap is his name. He's a zoologist, herpetologist. So we went into exact numbers and we said, hey, listen, there's only a few thousand snake species, lizard species. There's only like 10,000 bird species. And we literally, well, that was the debate. And we showed that based on the young Earth creation model and the rapid speciation that took place, the amount of species we actually have today of all these different species of animals, really, like if there's 4,000, I can't remember if it's the snakes or the lizards, if there's only, let's say 4,000, well, that's really only one new species a year. And you can read like specific predictions from creation scientists that are saying, okay, these snakes or these birds, this is how many per year since the flood would have to, like new species would have to form to explain since. Well, here's one question for you, actually, I guess, before we end it. And if you want to join the after show, you can too, or if you have some friends that want to join. But if there's, let's say there's between 10 and 12,000 bird species, okay, on the world today, you can Google it. That's how much there is roughly. That's only between 2 and 3 new bird species a year for 4,500 years since the flood. That's easily explainable, especially we see new bird species. Well, last year, we just seen a new species of finches on the Galapagos Island. And the only reason we see that is because we're actually studying it and observing it. Imagine all the different species that are forming around the world just because we're not even looking, like the Amazon and stuff like that. Well, that's easily explained, but why? If birds evolved from dinosaurs 65 million years ago, that's a long time ago, you know? Why is there only 12,000 bird species on the planet?
Well, there are more. It's just a lot of them went extinct. I mean,
right, and that is, you know, and I've heard that answer before. The question would be, or the next question would be, you know, have you, I know you haven't, but have any evolutionary scientists, and I don't believe they have that I know, made any specific predictions on extinction rates, speciation rates? It's just like the mitochondrial DNA. If they say that mutation rates in the mitochondrial DNA were slower in the past, okay, make me a prediction. How fast or when does the mitochondrial DNA clock accelerate? When does it slow down? You know, I just don't want like post hoc ad hoc. I want actual predictions. So yeah, extinctions could, I mean, that'd have to be a lot of extinctions, of course, but if there can be like a model, a workable model with testable predictions as to the rate of extinction events, then that explanation could be science. As of now, it's kind of just
well, there's been a lot of extinctions. I mean, you know, we don't have any, you know, non-avian dinosaurs anymore. You know, we don't have the dodo bird. There's lots of things that have gone extinct.
Right. No, extinction obviously is absurd, and that would be according to our model. I mean, just look at how many billions of fossils we find. I mean, there's been a lot of extinction events, but I know there's a few guys that want to jump into like a post-show, after show, which means you're invited. Generally, in the after show, I'm going to let the other guys do most of the talking, which means if you want to join and just have a friendly back and forth with a few of the other guys, you're welcome to. That's all I want to give you. I'm going to give you the last word since you're the guest. I want to say, David, it's been extremely respectful, profitable. I enjoyed talking to you. We have a lot in common, which is awesome. Your answers were great. Your questions were great. The point is, hopefully we made each other think. But yeah, if you want to just have the last word, give your concluding statement. I won't say anything afterwards. And then we'll have Praise, the producer. He'll do like an after show, and you can join. You know, we like you, so we're happy to have you anytime, David. So take it away, brother.
Thank you so much. Yeah, y'all are awesome. I really enjoyed talking with you. You've really opened up my mind too. Also, yeah, I've got a lot of work to look up from now going forward. Yeah, there's a lot of stuff that I don't know about that I need to research and address. And thank you so much for bringing that to my attention. And also, I wanted to apologize to Nephilim Free. I emailed him and asked him if he wanted to debate me on evolution, and we did eventually agree to a date and time, but then immediately, me being, I don't know why I did this. I asked him if he would be okay with Aaron Raw appearing in the debate, and in Nephilim Free, he responded and said that he's been, wanting to debate Aaron, but Aaron won't, agree to debate him in a scenario like this. That was not appropriate of me to ask. So I want to apologize to Neff.
Oh, that's not a problem, Fran. The problem is that Aaron Raw wants to yell and talk over somebody and berate them constantly. If you're in a hangout where he and a friend or two of his have control over it, but he won't debate in a structured debate where people are timed, because he doesn't want the creationists to be allowed to finish an assertion or an argument, because he fears that the information they give will refute him in the public eye, and he doesn't like that. So he only wants to engage people where he can berate them, yell over them, and talk over them. And if it's not a structured debate, I'm not interested in debating with him, because that's all it would be. Him talking over me constantly and me not being able to finish a sentence, So structured debate's what I want, but he won't do that. And so it's all good,
Thank you.
It's all good.
Especially because not everybody is as respectful and easygoing as you, David. So sometimes it's like Neff was said, structured formal debate is, especially with someone like Aaron, it would be the best way to.
Yeah, with some people it's necessary because it won't work any other way.
Exactly. But Praise, if you're there, my recommendation would be, just so this specific debate's not 4 hours, maybe we'll end this since this is the debate. Praise recommended he'll start just like an after show, a separate stream where Neff, and I know there's a few other people that want to join. David, you're welcome to join if you want to have, if maybe you have some questions for Neff, I'd like to listen to that. I'll generally, since just spoke for the last couple hours, I'll let you guys kind of have a little discussion. So are you cool with that, Praise?
No.
Yeah,
I'm fine.
Yeah. So to the audience, hope everybody had fun. David, I really enjoyed this discussion. Both, I think, good points and good questions on both sides. We can reconvene in a month or so when you study some of the topics that you're interested in looking into. But yeah, I want to thank you for joining us. Like I said to the audience, Salvador, anybody who wants to join the after show, Praise is going to make it. He'll send out the links, and we'll chit chat for however long we can. So thanks so much, David. Thanks so much, guys.
Thank you so much, y'all. Y'all are awesome.
See you in a few minutes, chat.